Yeah, can everyone hear me?
Yes, sir.
Okay, sure. Okay, so in this, I have a list of sample problems. So each of these are MCQs, the same pattern in which you will have for your final exam. We, for most of these, we can use the idea of elimination method. One can try to justify why a particular answer could be incorrect. You can raise your hand and then we can go ahead with the discussion. So in this week's class, majorly the topics that were taught include growth curve, counting of cells, um, and a few other aspects with respect to carbon metabolism. So yeah, so the first question is which of the following is the cofactor for hexokinase? So if anyone would like to answer first, I want the person to uh, state what hexokinase is, in which pathway it is a part of, and then move on to the cofactor. That way it will help everyone. Yeah, Ramesh, you can unmute yourself. Yeah, you can unmute yourself, Ramesh.
Mg2 plus.
Yeah, first you can tell us what hexokinase is, which part, pathway it is a part of, and then we'll go to the cofactor.
Sound taking come. Sir, once repeat the question.
So I was telling, in order to answer this question, you can start with what hexokinase is, what is the substrate, what is the product that you get, and which pathway it is a part of, and then we can move on to the cofactor, so that
So
we have. Hmm,
regarding the glycolysis pathway,
hmm,
glucose converted into glucose 6-phosphate, which are catalyzed by the enzyme hexokinase.
okay, fine. Uh, Also, does this have any substrate specificity? Can only glucose be
No, each can
converted?
act. Hexose it can act as any kind of monosaccharide, either it may be glucose, fructose, mannose, or galactose.
Right. As long as it's a six carbon,
Yeah, 6 carbon containing compounds.
yes, clear. Yeah. So, and which step does this act on? Is it the first step or
First step on regarding the phosphorylation process.
right? And do you also know the significance of this? Why should this hexose be phosphorylated? Any idea about that?
No idea regarding the significances.
Okay, fine. Thanks. You can mute yourself.
Okay, sir, thank you.
Yeah. So, like he had mentioned, this is a part of glycolysis. This is a part of glycolysis where it takes in ATP, converts that to ADP, and adds the phosphate group to the hexose molecule. In glycolysis, the hexose molecule is glucose. And with respect to the biological significance, converting glucose to glucose 6 phosphate helps in transport, thereby driving the reaction forward. We'll go to the next question. In glycolysis, two ATP molecules are utilized by the enzymes dash and dash. Option A hexokinase and pyruvate kinase. Option B enolase. Option C pyruvate kinase. Option C uh, hexokinase and phosphofructokinase. and option D, aldolase and enolase. So with respect to this question, if anyone would like to answer, you can start with eliminating options. If option D is incorrect, you can justify why option D is incorrect. So multiple people can try to attempt. Each person can try to eliminate one particular option. That would also be helpful. And this session is primarily meant to be interactive, which is why it's expected that the students here communicate. We'll wait for 2 more minutes, Ramesh, and then we'll uh, start discussing.
So option C is correct.
So we have to eliminate. So you can give an incorrect option and try to justify why that option is incorrect.
Okay, okay. I
That'll
choose
help.
incorrect only.
Incorrect option and try to justify why that is incorrect. You can
So
talk about
option
one.
A is incorrect.
Okay, you can try to justify why that is incorrect.
So in case of hexokinase, it do utilizes ATP, but in case of pyruvate kinase, we yield ATP. We
Okay.
get ATP.
Okay, so now because we are discussing enzymes, it's always helpful to discuss about the substrate and product, what reaction a particular enzyme is carrying out. Earlier we discussed that hexokinase, from the name itself, it's kinase means it would add a phosphate, meaning it will take some phosphate source, some triphosphate. It could be ATP or GTP. So we know that hexokinase is the right answer, but pyruvate kinase also shows that it takes in it could also phosphorylate a moiety, right? So when you're talking about these options, when you're trying to eliminate these, talk about the substrate and the product that you get when then enzyme is used. So you were
Sir,
saying 1 minute. I think it was Mona Lisa. Yeah, you can uh, try to say why you
Uh,
meant pyruvate kinase is wrong.
yes, sir. In case of pyruvate kinase, we used to convert the phosphoenol pyruvate to pyruvate by using the pyruvate kinase. But in case of hexokinase, we used to convert the glucose into glucose 6 phosphate by the help of hexokinase.
Okay, so you're saying that is incorrect.
D is
Yes, sir.
incorrect.
Okay, and Ramesh, you were saying D is incorrect. Okay, what
D
does
is
an
incorrect.
B or D?
D.
Okay, what does an aldolase do?
So aldolases can act as a splitting of fructose 1,6 bisphosphate to DHA and glycerol 3 phosphate.
Right,
So that is only cleavage mechanism.
right.
I mean
And the
cleavage reaction.
hmm,
Regarding the enolase is a hydration process. No need of any ATP.
hmm.
So it's only
And
one.
is enolase a part of glycolysis?
Yeah.
Hmm,
The conversion in between the 2-phospho glycerate to phosphoenol pyruvate.
hmm. Okay, so we've eliminated options A
D.
and D. Okay, if anyone else would like to volunteer, that will also be helpful. Does anyone want to give this a try? Okay, Mona Lisa or Ramesh, any one of you can unmute and talk about the third option, but that is incorrect.
Yes, sir. Option B is also incorrect.
Okay. Okay, so now because we have discussed the incorrect options, the the right option, you can also talk about it and what is the substrate and the product that we get with each of the enzymes in the right option.
So option C is correct.
Okay. So what is the substrate for phosphofructokinase?
So for phosphofructokinase, it used to convert the fructose 6-phosphate to fructose 1,6-bisphosphate.
Right. And that process needs ATP. And hexokinase we've discussed earlier. Right. I hope that was clear to others. I would go to the pathway again. So if we look at these, there are in glycolysis, there are two particular steps where an ATP is required by a kinase. 1 minute. Yeah, there are two steps where an ATP is required by a kinase, which is the first step where hexokinase acts on glucose and converts it to glucose 6-phosphate. And then phosphofructokinase acts on fructose 6-phosphate to give fructose 1,6-bisphosphate. So these are the two reactions where ATP is required. The next question is fructose 1,6-bisphosphate is broken down into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by option A enolase, option B aldolase, option C pyruvate kinase, option D isomerase. Right. We can take a couple of minutes and then try to answer this question. Since this session is supposed to be interactive, it would be helpful if others would also interact.
Shall I, sir?
We can wait for a couple of minutes. I just want to see if others would also like to attempt answering these questions. If not,
Okay.
I'll tell you, you can start explaining. Okay, Vivek. Huh. We'll wait for 2 more minutes. Okay, Vivek, you can talk about one incorrect option, whichever you would like to choose, followed by Isha.
Sir, option A is the wrong option, sir.
Oh,
Enolase.
okay, what does enolase do?
Enolase 2-phosphate glycerate convert करते हैं sir to phosphophenol pyruvate by removing water molecule.
Okay. Hmm. Yeah, you can mute yourself. Huh. Isha, you can talk about another incorrect option.
Sir, option C, pyruvate kinase was incorrect because they convert phosphophenol into pyruvate. That's why they are incorrect.
Phosphophenol, is it?
Pyruvate kinase C number is incorrect.
Yeah, yeah. What is the substrate in the product there when you have pyruvate kinase? Pyruvate kinase?
The pyruvate kinase converted into pyruvate. The last.
Hmm. Okay. Ramesh, do you want to go for the other incorrect option?
Isomerase.
Okay. What isomerase do we have here in glycolysis?
Actually, the isomerization reaction takes place in glucose 6-phosphate to fructose 6-phosphate,
Hmm.
glucophosphoisomerase.
Hmm. So can we
And
call it
and
a
then
hexose isomerase?
yeah.
Right.
And one more regarding the glycerol 3-phosphate and dihydroxysteine phosphate,
Uh-huh.
having one more isomerase is there, triose phosphoisomerase.
Hmm.
So actually, my intention is isomerase is the wrong answer.
Okay. Right. So, like it was mentioned earlier, enolase converts 2-phosphoglycerate to phosphoenol, phosphoenol pyruvate, whereas the two isomerases that are given, because this question did not provide which isomerase it's asking for, we have to think about both. One is the phosphohexoisomerase, which converts glucose 6-phosphate to fructose 6-phosphate. We are looking at the step where fructose 1,6-bisphosphate is converted to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The other isomerase that's there is the phosphotriose isomerase, which was told earlier. This converts glyceraldehyde 3-phosphate and dihydroxyacetone phosphate into each other. The enzyme responsible for breaking down this fructose 1,6-bisphosphate into G3P and DHAP, which are these, is called aldolase. So the right answer is option B. Huh. So the next question is, which of the following cell surface receptor is involved in the uptake of glucose? Option A, FGF, option B, EGF, option C, GLUTS, and option D, insulin receptor. We'll take a couple of minutes and then you can raise your hands. Okay. Yeah, Vivek, you can talk about one incorrect option and justify why that option is incorrect.
Sir, epidermal growth factor is the wrong option, sir.
Okay.
Because it is a peptide growth factor, sir, that promotes only cell growth and differentiation.
Hmm. So
So it's
option.
not involved in glucose uptake.
Hmm. And one more question: can growth factors act as receptors, or do they bind to receptors?
Sir,
Can
sir,
growth
can you
fact
repeat it again, please?
Yeah, can growth factors act as receptors, or do they bind to receptors?
Mm. So no, sir, growth factors cannot act as receptors, sir.
Okay. Hmm. Fine. You can mute.
Okay.
Right. Uh, yeah, Ramesh.
Sir,
Yeah, you can talk about one incorrect option.
so I go with actually right correct answer. I know, sir. Incorrect means actually FCF. A is a incorrect answer, but what is the reason I don't know.
Okay.
I know
So
I know only for
do
the
you know what is FGF?
uh, uh, what, sir?
Do you know what is FGF?
Yeah, FGF means abbreviation. I don't know, sir. Actually,
Okay.
FGF means
It's another growth factor, fibroblast related growth factor.
okay, okay, fibroblast growth factor.
Yes.
Uh,
Right. Okay, so what about insulin receptor then? Why is
so
what do you think about that? Does it also bind and help in uptake of glucose?
So whenever if any excess amount of cyclic AMP present in our cells,
Hmm.
uh, that time the insulin receptor to play a major role uptake of glucose. One is that one,
Okay.
and GLUT means glucose transporter 4 also play a predominant role, especially for uptake of glucose through cell membrane.
Hmm.
The two only right answers actually, but my
Okay.
thought only, but not exact.
Okay. Okay. So with respect to this question, we can't consider we can only consider option C, GLUTs. This has nothing
Okay.
but the glucose transporters. So as soon as insulin binds to its receptor in the cell membrane, what happens is this activated receptor it promotes for glucose transporters to be available on the cell surface. That is what is shown here. So these vesicles are going to the membrane and it fuses. And this vesicles contains these membrane bound transporters. And once these vesicles could fuse to the cell membrane, these transporters are available on the cell membrane. Now, depending on the level of glucose, one minute. Yeah, now depending on the level of insulin, glucose transport would be altered. So this is also explained in lecture 4. So the right answer is GLUTs, the glucose transporters. Okay. Uh, the next question is, during the event of low blood glucose, the enzyme pyruvate kinases, option A, phosphorylated, option B, dephosphorylated, option C, ubiquitinated, and option D, glycosylated. So if anyone would like to answer, they can raise their hands. We'll take a couple of minutes and then start discussing this. Okay, does anyone want to attempt this question? So with respect to this question, you can talk about what the particular process would be, what is phosphorylation, what is dephosphorylation, ubiquitination, and glycosylation. And then we can proceed. Okay, if no one would want to try, I'll go through each option one by one. Option A is phosphorylation. So phosphorylation, this is a post-transcriptional mechanism where a phosphate group is added to a protein. The protein could be enzyme, and once this phosphate group is added, the enzyme starts functioning or gets activated. Whereas dephosphorylation is the removal of a phosphate done by a phosphatase, which would tell a particular enzyme to stop functioning in a particular manner. These are signals that come to proteins once they have been translated, and these do not come during translation itself. Third option is ubiquitination. So that is the addition of a smaller protein. And the fourth option is glycosylation. Glycosylation means addition of a glycosyl moiety. In many cases, it could be glucose. And glycosylation either could act as a way in which the cell conveys that there is a lot of glucose in the cell, because of which glycosylation is happening, or with respect to small metabolites like the hydrophobic plant metabolites, their glycosylation helps in improving the solubility of these metabolites so that they can be transferred in the hydrophilic environment. Now, with respect to pyruvate kinase, if we look at the regulation pathway of pyruvate kinase, this phosphorylated pyruvate kinase is less active, whereas the dephosphorylated pyruvate kinase is more active. This pyruvate kinase in general, it acts to give you the pyruvate, and it can be negatively regulated in the presence of higher concentrations of ATP. So with respect to pyruvate kinase, the answer here would be option A, which is phosphorylation. So if this is not clear, and if anyone has a question, I can repeat it. Otherwise, we could go to the next question. Okay, since there are no questions, we'll go to the next question. One molecule of GTP is equivalent to dash molecule of ATP. Option A 1, option B 2, option C 3, and option D 4. So here one can talk about where GDP plays a role with respect to the processes that we are discussing in, and then try to come up with why GTP could act as an equivalent of ATP and what is the stoichiometry of this equivalence. Okay, here in the Krebs cycle ATP balance sheet, we see that the total amount of ATP that is being produced directly or indirectly. So for every NADH, we get 3 ATPs that are being produced, and for FADH, we get 2 ATPs. In one particular step, we have this GTP that's being generated, which would also could be uh, equated to ATP because of the functional roles and their possible ability to phosphorylate. So, with respect to that, one molecule of JTP can be considered as an equivalent to one molecule of ATP. The next question is, which of the following pairs positively modulate the conversion of isocitrate to alpha ketoglutarate? So, would anyone like to unmute and talk about this particular step of isocitrate to alpha ketoglutarate production? Yeah, Vivek.
Sir,
So,
um,
yeah, you can. So, which pathways are we discussing here?
sir,
Isocitrate, alpha ketoglutarate,
Krebs cycle, sir.
Krebs cycle. Okay. Now, yeah, you can, like we earlier did, you can try to justify why a particular option could be wrong.
Sir, option A, ATP and NADH.
Okay, why do you think it's wrong?
Sir, exact reason, I'm not sure about the exact reason, sir.
Okay.
But both ATP and NADH will inhibit the isosaccharic dehydrogenase, sir.
Oh,
So
oh,
that's why maybe
so
that's the reason.
it'll negatively modulate. It will not
Yes,
positively modulate. Okay.
yes.
Okay. Any idea as to why ATP and NADH should negatively modulate?
No idea, sir.
Okay, you can think about these reactions as connected series of mechanisms. Where do you get an ATP?
Sir,
Where do we get ATP?
uh,
Okay. We'll talk about major reactions that contribute to production of ATP. So glycolysis is one pathway, right?
yes, sir.
Through which we get equivalents or reducing equivalents, which is the NADH, which then go to the mitochondria and then to give uh, ATP, like we previously saw the balance sheet. Now you can try to think why ATP would inhibit, or if that's difficult, it's okay. I'll try explaining that. So calcium can be considered
But
as a, sorry, yeah,
so yes, sir. Please.
yeah, you can, if you want to,
No, sir. I don't know the exact reasons, sir. So
okay. Okay, so for calcium can be considered as a cofactor, and in general, cofactors help in accentuating reactions, which is why it can be called as a positive modulator for the conversion of isocitrate to alpha ketoglutarate, this particular step. With respect to ATP and NADH, these are intermediates that, or not just intermediates, this is what you get from glycolysis. And and if these are consumed here, that would lead to lesser production. So biochemically, that would be uh, a disadvantage, because of which they do, do not allow for conversion of isoseparate and stop at a particular stage, which is why they are called as negative modulators. The next question is, production of alcohol under anaerobic conditions in yeast is facilitated by the enzyme alcohol dehydrogenase, which converts acetaldehyde to ethanol. The substrate pyruvate binds the active site of the enzyme with the help of a dash ion. So if anyone would like to answer this question, first you can try talking about what an anaerobic condition is and what it would lead to, uh, what secretion that it would lead to. Then we can go about discussing the alcohol dehydrogenase. You can raise your hand. Yeah, Chandrama, you can start with explaining what anaerobic conditions is and how would that lead to rearrangement or changes in the pathway flux.
Actually, an anaerobic condition is that condition in which there is absence of oxygen in the respiration.
Okay, so what would that lead to?
Which would lead to production of that to convert that pyruvate into ethanol,
Hmm.
or
And
it can also do the lactate formation.
yeah, lactic acid is a major uh, product that you get in anaerobic conditions. True. Hmm. So now, with respect to this question, what do you think is the right answer?
The correct answer is zinc, because for alcohol dehydrogenase we require zinc ion to bind to that its active site to activate.
Hmm. Right. So you're saying pyruvate. So you're saying pyruvate binds to the active site of this enzyme through zinc. Hmm. Yes. As this was also discussed in the course. So this is how the mechanism happens, and partial negative charge on this acetaldehyde oxygen helps in it binding to zinc. Now, because oxygen has a negative charge, now later this moiety, this NADH, loses this H. meaning it is a loss of proton, which is an oxidation process. Now then, this is converted into ethanol. So the right answer is option C, zinc. The next question is, which of the following is the function of phenol red in DMEM? Option A, regulate dissolved CO2. Option B, detect change in pH. Option C, enhance the activity of antibiotics. And option D, prevent the growth of bacteria. So if anyone would like to answer, they can raise their hands and talk about an incorrect option and what would help in playing the role of an incorrect option. First, we'll start with what DMEM is and what are the incorrect options. Whoever wants to answer can raise their hand. Yeah, Chandrama, you can talk about one incorrect answer.
That first option,
Okay.
regulate dissolved CO2. So phenol red's function is
You
not
need not
to
mention the role of phenol red, but you can talk about if regulating dissolved CO2 at all is an issue and what will help in that. What I'm asking is, okay, what is DMEM? DMEM is a medium for growth of particular organisms. Is that right?
Yes.
Okay, so now why should dissolved CO2 levels be regulated? Do you have an idea?
Sorry, sir, I am not able to hear that.
Okay, I asked you, why should the dissolved CO2 levels be regulated?
Because excess CO2 levels could lead to the cell die.
Why will a cell die if there is excess CO2?
Because the cell requires exactly oxygen for the respiration, not that CO2.
Yeah, that is there. So the cell would die for lack of oxygen, not for the presence of CO2. Do you think CO2, when dissolved in water, could alter the properties of water or the medium, in this case, DMEM medium?
It could make it acidic. It could
Right,
form carbonic acid.
right. So yeah, that's the answer I was looking for. Yeah, so when CO2 is dissolved, that would lead to changes in the pH. So pH has to be maintained at the optimum pH for growth. So what do you think is there in DMEM that would regulate the dissolved CO2? Or what can actually regulate the pH? Is there something in the buffer? Okay, so media contain buffers. Buffers could either be salts of weak acids or weak bases, where they can basically dictate the amount of protons in a solution, amount of free protons. So if there is dissolved CO2, it might change the medium into an acidic medium. which can be prevented by reducing the amount of free protons. And this can be taken care of by having a buffer, like phosphate buffer or tris buffer. So option A is incorrect. Does anyone want to try talking about any other incorrect option and what could be there in a media that would play the role of that particular option? Yeah, Jakin. Do you want to talk about a particular incorrect option?
Yes, sir. Sir, I think that like phenol red is more of an indicator thing.
Hmm.
It like it does not detect the dissolved CO2. Neither it detects the like activity of antibiotics, and it neither has any effect on the growth of bacteria. So I will go for option B because it changes the color, I guess.
So when does phenol red changes its color?
Sir, when it's like used in this thing called DMM, Eagle Medium Motifs,
Hmm.
it just changes color in response to like the pH of the color, pH of the medium, sorry.
Hmm.
Yep.
And what would prevent the growth of bacteria?
Um, I'm not sure, sir.
Okay, any generic option, general answer.
Sir, they are like antibiotics can prevent the growth of
Yeah.
bacteria.
Huh. And do you
Uh,
think if there are any molecules that can enhance the activity of antibiotics?
um, uh, I'm not sure, sir. Like they will just work on the wall,
Right,
NAM, all this
right,
stuff.
right, right. So the answer that you say is that phenol red plays the role of identifying if the or they change color with respect to changes in pH. That is right. And like you had mentioned, there is not a single enhancer that would improve the activity of antibiotics. So these antibiotics are small molecules, meaning the molecular weight is lesser than 1 kilodalton. So activity of antibiotics can be changed if the functional groups on the antibiotics would change, but not by any molecule like we see in the regulation pathways earlier. So the right option is option B, that it detects a change in the pH and changes its color. So it's an indicator. The next question is, in a Petroff-Hauser counting chamber, the volume of each chamber is approximately option A 10,000 inverse millimeter cube, option B 12,000 inverse millimeter cube, option C 20,000 millimeter inverse, and option D 40,000 inverse millimeter cube. Anyone who would like to attempt can raise your hand. If you don't know the right answer, that's fine. You can first talk about what is this Petroff-Hauser counting chamber, as it was discussed in the videos. Yeah, there's Anyone want to try answering what this Petrov-Hausser counting chamber would be? Yeah, Jakin.
Sir, I'm not sure about the answer, but like it's a counting chamber used to count cells, like on a slide that is designed for it in like blood hematological works and something like that.
So this can help in counting the number of cells,
Yes, sir.
right? Okay. Now, can this work at all densities of cells, or should any variation be made?
Yes, sir. Sir, I guess on the cell concentration can be calculated if that contravenes to density, because it just counts the total number of cells per slide, or there are some square areas that are approximated for it.
Hmm.
It can count. I think we can like count the or just approximate the density of the cell in relative measures.
Hmm.
Yes, sir.
Okay. Yeah. So, yeah, with respect to this question, this is not a question to think about, but this Petrov-Hausser counting chamber looks like this, where you add a little bit of your cells and put a glass slide. The volume is mentioned here as 20,000 inverse, which is option C here. Then, when observed with a microscope, so there are some divisions here. You can think of it as crosses. So in each of the crosses, you'll be able to count the number of cells. And if your cell suspension is uniform, ideally each of the chambers should have more or less an average equal number of cells, based on which we can count or get an approximate idea of the total number of cells per ml of bacteria.
Okay, sir.
Yeah, and there is another tool called hemocytometer, where you also have different chambers, or it has been partitioned into different chambers. There you take and you do an averaging calculation. Here you have a constant that would help you in determining or extrapolating the total number of cells. We'll talk about the constant here. Yeah. So now, if each chamber of this Petrov-Hausser counter has 5 bacterial cells, then the total number of cells per ml of the culture is. If anyone would like to answer this question, you can talk about the method that you have used. Yeah, does anyone want to try answering this question? Okay, so how do we do this is there is a constant of, okay, so we know that the volume of each chamber is 20,000 inverse millimeter cube. Now they're asking per ml, which is for centimeter cube. So if there are 5 bacteria cells, for a millimeter cube, you need to multiply it into 20,000. So 5 into this 20,000. That would give you 10 into, sorry, that would give you 10 power 5. Now that is for a millimeter cube. But if you want it for an ml, it's centimeter cube. So you multiply it by 1000 further, that gives you 10 power 8. So option C, 10 power 8 is the right answer. Now we move on to question 12. The correct sequence of different phases of bacterial growth is option A, stationary, followed by log, followed by lag, followed by death phase. Option B, lag, followed by log, followed by stationary, followed by death phase. And option C, log, followed by lag, followed by stationary, followed by death. And option D is lag phase, followed by stationary phase, followed by log phase, followed by death phase. Anyone who would like to answer can first talk about what each of the phases would be, and then we'll try to get the correct sequence. If anyone would like to answer, please raise your hand. Yeah, Vivek, you can talk about one particular uh, growth phase.
So, lag phase
Hmm.
is the initial phase where bacteria will be introduced into a new environment, sir.
Okay. What else do you know about it?
Uh, so, no cell division will occur in this phase. So,
Right.
the population will be remain stable.
Okay, when you say stable, does it mean that would grow linearly?
No, sir, the population will go same, though. I'm not sure about that, sir.
Okay, that's fine. So lag phase is the first stage where, because the cell is introduced to a particular new environment, like you had mentioned, it would take some time
Yes.
to grow.
Okay,
So
sir.
what you can understand about lag phase is the time when these would these cells would take more time to adjust to their surrounding environment, but they would continue growing, but
Okay.
but they won't have a higher growth rate.
Okay, sir.
Fine. Uh, does anyone want to talk about another growth phase? Chandrima? Yeah.
Log phase. In this phase, there is an exponential increase in the number of bacterial cells.
Okay. What is this phase? Uh, okay. What phase is log phase coming after?
Sorry, sir.
Okay. So there are these growth phases can be divided into many, right? So
Yes,
there
sir.
is some phase after which log phase or exponential phase happens. So what is this phase after which log phase happens?
Actually, before this is lag phase will be doing, and after that it is stationary phase.
Okay. So what happens during stationary phase?
In stationary phase, generally the old bacteria or the which has been first grown in the exponential or log phase, some of them get died due to overgrowth of that bacterial cells.
Okay, that doesn't happen exactly that way because this is binary division. Each cell gives rise to two cells, right? So there is no idea of this old phase or new phase. Sorry, old cells or new cells. At a given time, almost all the cells would have been generated by the same would have been generated at the same time. But what happens during stationary phase is that the amount of nutrients that is left in the media will start to decrease, which would lead to an overpopulation. So there will be an equal amount of cell division and cell death. So which is why in stationary phase, it's a flat line and there is no exponential increase like you had observed in the log phase. Uh, Followed by this is the death phase, where the number of deaths are more than the number of growth. So every bacterial growth goes through the stage where it starts with a log phase, where it is getting used to the environment and adapting to it, and then at the end of this lag phase, it starts growing because it has now adapted, it's comfortable to grow, and there is enough media for it to supplement its growth. But after some time, the population of cells increases and it would lead to more cells but lesser nutrients. So, cell division and cell death would happen at the same rate, leading to stationary phase, followed by death phase, where the number of active cells that are dividing will be lesser than the number of cells that are dying. So, you have a reduction in the cell density or cell population. The next question is, in which of the following phase, newly divided bacterial cells derive nutrition primarily from older dead bacteria in a culture? Okay, so if anyone would like to answer, you can try talking about this particular phase. Yeah, Jatin.
Sir, it might be the death phase, I guess, because uh, only it can derive the nutrition from the dead bacteria because like the other nutritional mediums might not be available, I guess.
In death phase, most of the bacteria would die. So you won't even have bacteria to derive or take up nutrition. That is a way in which you can understand the death phase. So we,
There is
if
a depletion of like nutrients in the
there
culture.
is depletion of nutrients, that's true, because of which almost all the cells would start dying, which is why the rate at which they die would be greater than the rate at which they divide. So
Right.
predominantly, it's all the cells are dead there. So you can't talk about death phase with respect to deriving nutrition there,
Fine, fine.
but you're close by. What other phases are there in the cell which is close to the death phase?
Sir.
Or in which other phase do you also observe death?
Sir, it can be like stationary phase because it's a kind of balanced thing. There are also new cells dividing at the same time and utilizing the nutrients. However, there is also like waste accumulation and there are also deaths in there.
Hmm, true. In the
It's a
stationary
kind of
phase,
stabilization.
yeah, sorry, can you repeat? It's kind of stabilization.
Yeah. And in the stationary phase, it's kind of stabilization. The death rates and nutrients are used. They are depleted in the way,
Hmm.
but these waste products they accumulate,
Hmm.
and there is also growth and death. So population kind of stabilizes.
Hmm. True. So, with respect to this question, you can consider stationary phase as the phase in which these cells can derive nutrition from dead bacteria, because here you have bacteria that are growing and bacteria that are dying, and both these rates are equal. And because these are equal, it also means that they still have the ability to divide. And because other bacteria are dying, they are source of nutrition, which is why stationary phase is more apt compared to death phase. In lag phase and log phase, you don't see this happening because the amount of nutrition that is available from the media is more than sufficient and it need not rely on dead bacteria. The next question is, bacterial culture medium was inoculated with roughly 5,000 E. coli cells at 10 a.m. and allowed to grow for 12 hours. Calculate the number of generations the cells have grown in the given time span, considering that there was no lag phase. Life cycle of E. coli is 20 minutes. So, if anyone would like to try, you can tell me uh, and the logic you use to calculate this. You can raise your hand if you are aware of it. We'll wait for a couple of minutes and see if someone else is able to answer. Otherwise, we'll go with Jakint and Chandrima. Okay. Jakint or Chandrima, anyone can unmute and talk about the right answer, how you calculated it.
Sir, so I think like the E. coli life cycle is 20 minutes, so it's 12 hour into minutes, it's like 720 minutes.
Hmm.
If we just divide by 720, we get like 36 generations.
Right.
Considering that there is no lag phase.
Hmm. Yeah, that's how you do it. For every hour, three generations is for every hour we expect three generations of E. coli, and in 12 hours there will be 36 generations, considering that there is only log phase and no lag phase. Right.
Yes, sir.
The last question is in the turbidimetric method, the OD of the bacterial culture is measured at option A 220 nanometer, option B 400 nanometer, option C 500 nanometer, and option D 600 nanometers. So anyone who would like to answer this question can first talk about what the turbidimetric method is, what an OD is, and then go to the wavelength. Yeah, Jarkant, you can first talk about what turbidimetric method is. Then
Yes,
Vivek,
sir.
you can talk about what OD is and what is the nanometer we can use for OD. Yeah, Jarkant.
Yes, sir. Sir, so like OD stands for optical density. It like it's a typical way of accessing the turbidity, the cloudiness of the sample, like how much bacteria or other particles are present in the current liquid culture under the observation. So
So,
when the light,
hmm,
yes,
yeah,
sir.
yeah, you can tell me.
So like when the light is passed through and it just shows the cloudiness, the suspensions of there is this scattering of light, and
Hmm,
I'm not sure about the nanometer, sir. Maybe 600 maybe.
okay.
Yep.
So that's okay. So you mentioned about scattering. What do you know about scattering?
Yes, sir.
Yeah, you can tell us what you know about scattering.
Sir, scattering is just like in our older books we used to test this experiment, scattering of light, and observe the sun rays through a forest, and the light just like the rays of light, they pass through the material, and but there is some materials are like cloudy in nature, or suspension materials.
Hmm. So scattering can be better defined as when particles are suspended, instead of absorbing, they reflect light in different directions because of the size of the particle. Can that be a better definition or better
Yes, sir.
explanation? Okay.
Yep.
Okay. Now, who was it? Vivek. Yeah. Vivek, do you want to try answering why a particular option would be incorrect here? Any option that you think would make sense as incorrect? Vivek?
Yes, sir.
Yeah. Do you think Okay, is there any way you can justify why a particular option could be incorrect here among the given options?
I'm not sure that's about this.
Okay, fine. Okay, so how we can look at this is any wavelength that is used for any of these experiments should not be absorbed. So you measure this OD, which is optical density or optical dispersion, on the basis of an incident light that has a particular intensity. When this particular intensity incident light falls on particles, it gets scattered in different directions, and we look at scattering in a particular angle. Now we also look at the intensity of the light that is being scattered. So we expect that the solution doesn't necessarily absorb lot of the light. Whatever goes in should maximally come out. So if you consider that there are bonds within biomolecules like proteins that can absorb at 220 nanometers, and some organic moieties can also absorb at around 400 nanometers and 500 nanometers, which leaves us 600 nanometers left. So 600 nanometers, there is this inverse relationship between energy and wavelength. So 600 nanometers being a higher wavelength would have very low energy, which is not of much significance with respect to molecules to transition from lower to higher energy. So this cannot be absorbed by lots of molecules, which is why OD is done at 600 nanometers. Yeah, so with this we come to the end of this question discussions. If there are any questions, you can post that here. We can discuss that. Also, if you would like to discuss, have you submitted assignment 1 on the portal? Anyone can answer if they have submitted and if the deadline is over.
No, sir. I
Okay, so
haven't
you,
yet.
okay, so the deadline is still on. Okay, fine.
Yes, 7 hours is the deadline, I guess.
7
Yeah,
hours.
7 hours.
Okay, fine. Okay, sure. Fine. So with this, we come to the end of week 2's sample problems. If there are any questions that you would want to discuss or any topics, we can try discussing them.
Sir, did I miss, I joined late in the lecture. Was there something I need to know? Or,
So this presentation and this video will be uploaded if
okay.
so where you can refer to the earlier sample questions. Which question did you were you when did you join? At which question?
Sir,
We
it
had
was
about
like,
15 questions.
yep. Sir, I think you were explaining the Krebs cycle thing.
Here is it?
Yeah,
Oh, Krebs cycle is below. Okay, here.
yeah. I joined here.
Okay, fine. So we were discussing a few questions with respect to glycolysis. Like the first one was, what's the cofactor for hexokinase? So are you aware of what hexokinase is?
Sir, it's an enzyme that break down, I guess.
Okay, so how you can, to understand enzymes, you can understand from the name itself. Hexo means six. So it
Six,
acts
yes.
on a six carbon moiety. And
Six
kinases
carbon.
are phosphorylating molecules. So this can phosphorylate a six carbon molecule. So this can either phosphorylate glucose or fructose, any six carbon molecule. Now they
Yeah.
ask you what is the cofactor of this hexokinase. So with respect to glycolysis, the cofactor that is needed is magnesium.
Okay, sir.
Then, yeah, then we looked at where ATP is used. in this process of glycolysis, which all enzymes need ATP. So if you look at the pathway of uh, this uh,
Mm. Glycolysis,
glycolysis,
right?
you observe that there are three kinases. One is a hexokinase, one is a phosphofructokinase, sorry, four
Right.
kinases. One is a phosphoglycerate kinase and the other is pyruvate kinase. Here,
Right.
in two of the processes, phosphate from the substrate is being converted and added on to an ADP to yield an ATP. So these two can be eliminated, which leaves us with two other options, hexokinase and phosphofructokinase. So option A, hexokinase and phosphofructokinase are the right answers.
Okay, sir.
Yeah. And the third question is fructose 1,6 bisphosphate is broken down into glyceraldehyde 3-phosphate and dihydroxyacetylene phosphate by. So this is a very straightforward question. Here we observe that aldolase breaks the 6-carbon moiety into 3-carbon and 3-carbon moiety. So that is the expected answer, followed by the cell surface receptor that's involved in uptake of glucose. So here the options that were given include FGF, EGF, insulin receptor, and GLUTs. So FGF and EGF are growth factors. One is the fibroblast growth factor, the other is the embryonic growth factor. Whereas GLUT-S and insulin receptors are either receptors or transporters. Now we are talking about a cell surface receptor that's involved in the uptake of glucose.
Glucose, sir.
So what happens here is, as soon as insulin is able to bind to its receptor on the cell membrane, it would allow for a few transport of few molecules. These molecules are nothing but glucose transporters, and these glucose transporters are bound to the membrane. That is how they can transport glucose into the cell. So there is a process that is involved here. First, these glucose transporters are placed on vesicles, which are also membranous. This happens as soon as this insulin binds to the receptor on its membrane. So these vesicles containing the glucose transporters then go and they reach the membrane, they fuse with the membrane. As soon as they fuse, the transporters are present at the interface of cytoplasm and the external source. So now glucose can be taken into cell. So the right answer is GLUT-S, which is glucose transporters. And the next question was relating to the regulation of pyruvate kinase with respect to the presence of different concentrations of glucose. So they asked about whether pyruvate kinase will be phosphorylated, dephosphorylated, ubiquitinated, or glycosylated. All of these are different post-translational mechanisms. So by post-translational, what we mean is that after translation, after the enzyme is produced, these processes, either addition of phosphate to a particular residue, or removal of phosphate from a particular residue, or addition of a protein called ubiquitin, or addition of a glucose moiety called glycosylation. Now, with respect to pyruvate kinase, what happens is it can undergo a cycle of phosphorylation and dephosphorylation. And pyruvate kinase, which is phosphorylated, is less active compared to the pyruvate kinase that is not phosphorylated. If the pyruvate kinase is not phosphorylated, it can convert more flux of phosphoenol pyruvate to pyruvate. Uh, Now they were asking during the event of low blood glucose, what would happen? So, if there is low blood glucose, the ability to produce pyruvate itself will be less. So it will be in this stage. of phosphorylated pyruvate kinase. As soon as you have more glucose, you would have more of phosphoenol pyruvate, which has to be converted to pyruvate. So there it will become dephosphorylated. And then we were looking at the reducing equivalent and the amount of ATP that is being produced, and it's equivalent to GTP. So this is where we were looking at the uh, ATP balance sheet. So here, all those steps where you have three molecules shows that one NADH has been transferred to mitochondria, whereas this step particularly showing two molecules shows that FADH has been transferred to mitochondria and plays the role of reducing equivalent. And in a particular step, this GTP is generated. One GTP can be considered as one ATP since both of them are triphosphorylated, and it's the phosphate that plays a major role here. After which we had gone to this Krebs cycle question, which you were there. So these are the questions that were discussed.
Yes, sir.
Right.
Okay, thank you, sir, for the recap.
Sure, thanks. If there are any specific questions, you can unmute and ask me, even with respect to week one's assignment problems. Yeah, we have about 15 minutes. Yeah, Jack.
Sir, I just wanted to ask, like, you explained the GLUT receptors and their role in, like, for the uptake of glucose, right?
Yeah.
So, but in the case of like excessive, there is more sugar, glucose present in hyperglycemic, like patients of diabetes and thyroid. So, like the function gets impaired.
Yeah, so
Of
there are, I am not very aware of the hyperglycemic conditions, but yeah, you can first ask me the question. I'll try to answer if I know about it.
yes, sir. I just wanted to know how like the insulin receptors and all this mechanism can be affected in the presence of less or hypo or hyperglycemic conditions, if that can be.
Okay. So in usually these, I know the chemistry behind these. I know the general chemistry as to how these enzymes would act and how they would get saturated over time, but I'm not very sure about the mechanisms that you're asking about.
Okay,
But
sir.
with respect to this, what I think is at that higher concentrations, these enzymes would saturate and no longer be able to do the required biochemical functioning, making them inactive, even if they are present. So that is possibly one method, or we can look at the specific method. Let me try to share my screen. 1 minute.
And so maybe the some diabetic patients pass sweet smelling like urine and all this stuff because the uptake they are saturated the GLUT receptors.
1 minute. Let me share my window. Yeah. So you're talking about insulin resistance. GLUT receptor, insulin. Okay, I'll add a.
Yes,
Oh,
sir.
1 minute. It's my. Yeah. I'll add a link to the PowerPoint presentation of a paper which you can try looking into. It's this paper where that has been referenced for this particular line, which talked about incorporation of Food4 into the membrane receptor and how this regulation happens. This talks about the signal transduction pathway, but not specifically with respect to the insulin resistance. But there should be references in this paper for insulin resistance. Let's see. Insulin.
Okay, sir. Sir, I guess like it's the same thing as you explained. Like there comes a
Hmm.
saturation point at which they cannot like catch the glucose molecules for reabsorption, and the blood is displaying this sweet odor of
Hmm.
like there is the fast movement in the nephron tubules, um, and the glut receptors cannot like catch the balls very fastly and get them reabsorbed. I'm like definite about this thing, what happens at hyperglycemic conditions, but I
Hmm.
am not sure, however, at the hypoglycemic ones, if there is low insulin or insulin resistant ones. That I will look up to, sir, the paper you mentioned.
Okay, sure. I'll upload the PPT. You can check that.
Sure, sir.
Okay. Okay. If there are no more questions, you can leave the class. Thank you for joining. The next class will be on Monday at 6 to 8 again.
you.